Teresa Pritzker, PhD, using a computer in her office.
My research focuses on building computational method to address problems in biomedical sciences.
Text, Teresa Pritzker, PhD, Senior Investigator, Computational Biology Branch, NCBI.
We are particularly interested in developing new methods to study diseases.
Graphic compares cellular elements for healthy versus cancerous mouse tissue.
This particular interest actually in cancer. Before, people have been looking at diseases from the perspective, okay, here's a gene, something wrong happened to this gene, and you have a disease. But complex diseases does not work this way.
Animation of blue DNA strand breaking apart.
Usually something happened to this pathway, something happened to this process, and when something happened to this process, you have a disease.
Spreadsheet shows genetic dataset. Chart shows interconnected data elements.
It's not so easy to identify the pathway when you look gene by genes, because it may be that every individual perturbation, when you look from gene by gene's perspective, is very small and it's hard to capture. But when you understand that you are to look at the pathway in group of genes, the signals get amplified, and you may be able to identify processes that you wouldn't see if you would look at each genes in isolation. So we are trying to look at the diseases from the perspective of dysregulated pathways.
Split screen shows red blobs of cancerous tissue on left and female patient in bed on right. Then left side shows drug capsules resting on document titled Cancer.
Some of our recent studies in cancer research are related to understanding of the relationship between mutations patient may have and, say, drug response.
Split screen. At left, Teresa working on her computer. At right, graphic titled Candidate Subnetworks Associated with Drug Sensitivity.
Assume that we identify a pathway, we can ask the question, does the patient has a mutation in this pathway? And this gives us some way of predicting whether or not the individual will respond to drug. Those questions are indeed very much driven by personalized medicine.
Graphical chart of mutation signatures. Computer monitor shows NCBI's RepairSig software screen.
I think the goal would be some of the methods that I've developed will have a really lasting effect, and also that some of the biological finding would be important for human health.
Animation of blue DNA strand behind text. Dr. Pritzker's research focuses on developing computational methods for systems biology, including application to cancer research, gene regulation, and methods for analysis of new types of experimental data. For more NLM research, visit nlm.nih.gov/research. NIH NLM gray and blue logo with underlining text, serving scientists and society.